Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Organelle. 87 languages. Organelle. Details. Pronunciation /RNL/. Part of cell. Identifiers. Latin organella. Mesh D015388. THH1.00.01.0000009. FMA 63832. Anatomical terms of microanatomy. Edit on Wikidata. Cell biology. Animal cell diagram. In cell biology, an organelle is a specialized subunit, usually within a cell, that has a specific function. The name organelle comes from the idea that these structures are parts of cells, as organs are to the body, hence organelle, the suffix "l" being a diminutive. Organelles are either separately enclosed within their own lipid bilayers, also called membrane bounded organelles, or are spatially distinct functional units without a surrounding lipid bilayer, non-membrane bounded organelles. Although most organelles are functional units within cells, some function units that extend outside of cells are often termed organelles, such as cilia, the flagellum and arculum, and the trichocyst. These could be referred to as membrane-bound in the sense that they are attached to, or bound to, the membrane. Organelles are identified by microscopy, and can also be purified by cell fractionation. There are many types of organelles, particularly in eukaryotic cells. They include structures that make up the endomembrane system, such as the nuclear envelope, endoplasmic reticulum, and Golgi apparatus, and other structures such as mitochondria and plastids. While prokaryotes do not possess eukaryotic organelles, some do contain protein-shelled bacterial microcompartments, which are thought to act as primitive prokaryotic organelles. 1. And there is also evidence of other membrane-bounded structures. 2. Also, the prokaryotic flagellum which protrudes outside the cell, and its motor, as well as the largely extracellular pilus, are often spoken of as organelles. History and terminology, edit. In biology, organs are defined as confined functional units within an organism. 3. The analogy of bodily organs to microscopic cellular substructures is obvious, as from even early works, authors of Explore Our Developer-Friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. Respective textbooks rarely elaborate on the distinction between the two. In the 1830s, Félix Dujardin refuted Ehrenberg theory, which said that microorganisms have the same organs of multicellular animals, only minor. 4. Credited as the first, 5, 6, 7, to use a diminutive of organ. i.e., little organ, for cellular structures was German. Zoologist Carl August Moebius, 1884, who used the term organula, plural of organulum, the diminutive of Latin organum. 8. In a footnote, which was published as a correction in the next issue of the journal, he justified his suggestion to call organs of unicellular organisms organella since they are only differently formed parts of one cell, in contrast to multicellular organs of multicellular organisms. 8. 9. Types, edit. While most cell biologists consider the term organelle to be synonymous with cell compartment, a space often bounded by one or two lipid bilayers, some cell biologists choose to limit the term to include only those cell compartments that contain deoxyribonucleic acid, DNA, having originated from formerly autonomous microscopic organisms acquired via endosymbiosis. 10. The first, broader conception of organelles is that they are membrane-bounded structures. However, even by using this definition, some parts of the cell that have been shown to be distinct functional units do not qualify as organelles. Therefore, the use of organelle to also refer to non-membrane-bounded structures such as ribosomes is common and accepted. 11. Verification needed. 12. 13. This has led many texts to delineate between membrane-bounded and non-membrane-bounded organelles. 14. The non-membrane-bounded organelles, also called large biomolecular complexes, are large assemblies of macromolecules that carry out particular and specialized functions, but they lack membrane boundaries. Many of these are referred to as proteinaceous organelles as their main structure is made of proteins. Such cell structures include large RNA and protein complexes, ribosome, spliceosome, volt. Large protein complexes, protosome, DNA polymerase III holoenzyme, RNA polymerase II. Holoenzyme, symmetric viral capsids, complex of GRO-EL and GRO-ES, membrane protein complexes, porosome, photosystem Y, ATP synthase. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Large DNA and protein complexes, nucleosome. Centriole and microtubule organizing center, MTOC. Cytoskeleton. Flagellum. Nucleolus. Stress granule. Germ cell granule. Neuronal transport granule. The mechanisms by which such non-membrane-bounded organelles form and retain their spatial integrity have been likened to liquid-liquid phase separation. 15. The second, more restrictive definition of organelle includes only those cell compartments that contain deoxyribonucleic acid, DNA, having originated from formerly autonomous microscopic organisms acquired via endosymbiosis. 10, 16, 17. Using this definition, there would only be two broad classes of organelles, i.e. those that contain their own DNA, and have originated from endosymbiotic bacteria. Mitochondria, in almost all eukaryotes. Plastids, 18, for example in plants, algae, and some protists. Other organelles are also suggested, by whom, to have endosymbiotic origins, but do not contain their own DNA. Citation needed, notably the flagellum. See evolution of flagella. Eukaryotic organelles, edit. Eukaryotic cells are structurally complex, and by definition are organized, in part, by interior Compartments that are themselves enclosed by lipid membranes that resemble the outermost cell membrane. The larger organelles, such as the nucleus and vacuoles, are easily visible with the light microscope. They were among the first biological discoveries made after the invention of the microscope. Not all eukaryotic cells have each of the organelles listed below. Exceptional organisms have cells that do not include some organelles, such as mitochondria, that might otherwise be considered universal to eukaryotes. 19. The several plastids including chloroplasts are distributed among some, but not all eukaryotes. There are also occasional exceptions to the number of membranes surrounding organelles, listed in the tables below. For example, some that are listed as double membrane are sometimes found with single or triple membranes. In addition, the number of individual organelles of each type found in a given. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cell varies depending upon the function of that cell. The cell membrane and cell wall are not organelles. Major eukaryotic organelles. Organelle main function structure organisms notes. Chloroplast. Plastide. Photosynthesis. Traps energy from sunlight. Double membrane. Compartment. Plants, algae. Rare. Kleptoplastic organisms. Has own DNA, theorized to be engulfed by the ancestral archaeplastide cell, endosymbiosis. Endoplasmic reticulum. Translation and folding of new proteins, rough. Endoplasmic reticulum. Expression of lipids. Smooth. Endoplasmic reticulum. Single membrane. Compartment. All eukaryotes. Rough endoplasmic reticulum is covered with ribosomes, which are bound to the ribosome membrane, has folds that are flat sacs, smooth. Endoplasmic reticulum has folds that are tubular. Flagellum locomotion, sensory protein. Some eukaryotes. Golgi. Apparatus. Sorting, packaging. Processing and modification of proteins. Single membrane. Compartment. All eukaryotes. CIS face, convex, nearest to rough endoplasmic reticulum, transface. Concave, farthest from rough endoplasmic reticulum. Mitochondrion. Energy production from the oxidation of glucose substances and the release of adenosine triphosphate. Double membrane. Compartment. Most eukaryotes constituting element of the chondriome has own DNA, theorized to have been engulfed by an ancestral eukaryotic cell. Endosymbiosis. 20. Nucleus. DNA maintenance controls all activities of the cell. RNA transcription. Double membrane. Compartment. All eukaryotes contains bulk of genome. Vacuole storage. Transportation helps. Single membrane. All eukaryotes Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Maintain homeostasis. Compartment. Minor eukaryotic organelles and cell components. Organelle / macromolecule main function structure organisms. Acrosome. Helps spermatozoa fuse with ovum. Single membrane. Compartment. Most animals, including sponges. Autophagosome. Vesicle that sequesters cytoplasmic material. And organelles for degradation. Double membrane. Compartment. All eukaryotes. Centriole. Anchor for cytoskeleton. Organizes cell division by forming spindle fibers. Microtubule. Protein. Animals. Cilium. Movement in ORAV. External medium. Critical developmental signaling pathway. 21. Microtubule. Protein. Animals, protists, few. Plants. Cnidocyst stinging. Coiled hollow. Tubule. Cnidarians. Icepot apparatus. Detects light, allowing phototaxis to take place. Green algae and other. Unicellular. Photosynthetic. Organisms such as euglenids. Glycosome carries out glycolysis. Single membrane. Compartment. Some protozoa, such as trypanosomes. Glioxysm. Conversion of fat into sugars. Single membrane. Compartment. Plants. Hydrogenosome. Energy and hydrogen production. Double membrane. Compartment. A few unicellular eukaryotes. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Lysosome. Breakdown of large molecules, for example, proteins, plus polysaccharides. Single membrane. Compartment. Animals. Melanosome pigment storage. Single membrane. Compartment. Animals. Mitosome. Probably plays a role in iron-sulfur cluster, Fe(S) assembly. Double membrane. Compartment. A few unicellular eukaryotes that lack mitochondria. Myofibril myocyte contraction. Bundled filaments. Animals. Nucleolus pre-ribosome production. Protein DNA. RNA. Most eukaryotes. Ocelloid. Detects light and possibly shapes, allowing phototaxis to take place. Double membrane. Compartment. Members of the family. Warnaulaceae. Parenthesum not characterized. Not characterized. Fungi. Peroxism. Breakdown of metabolic hydrogen peroxide. Single membrane. Compartment. All eukaryotes. Porosum secretory portal. Single membrane. Compartment. All eukaryotes. Protosome. Degradation of unneeded or damaged proteins by proteolysis. Very large protein complex. All eukaryotes, all. Archaea, and some. Bacteria. Ribosome, ATS. Translation of RNA into proteins. RNA protein all eukaryotes. Stress granule mRNA storage, 22. Membranellis. mRNP. Complexes. Most eukaryotes. Tiger domain mRNA encoding membranellis most organisms. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A. Electron micrograph of Halotheobacillus. Neapolitanus cells, arrows highlight carboxysms. B. Image of intact carboxysms isolated from H. Neapolitanus. Scale bars are 100 nm. 23. Structure of Candidatus brucadia. Animoxidans, showing an animoxosome. And intracytoplasmic membrane. Proteins. Vesicle material transport. Single. Membrane. Compartment. All eukaryotes. Other related structures. Cytosol. Endomembrane system. Nucleosome. Microtubule. Prokaryotic organelles, edit. Prokaryotes are not as structurally complex as eukaryotes, and were once thought to have little internal organization, and lack cellular compartments and internal membranes, but slowly, details are emerging about prokaryotic internal structures that overturn these assumptions. 2. An early false turn was the idea developed in the 1970s that bacteria might contain cell membrane folds, termed mesosomes, but these were later shown to be artifacts produced by the chemicals used to prepare the cells for electron microscopy. 24. However, there is increasing evidence of compartmentalization in at least some prokaryotes. 2. Recent research has revealed that at least some prokaryotes have microcompartments, such as carboxysms. These subcellular compartments are 100–200 nm in diameter and are enclosed by a shell of proteins. 1. Even more striking is the description of membrane-bounded magnetosomes in bacteria, reported in 2006. 25, 26. The bacterial phylum Planktomystota has revealed a number of compartmentalization features. The Planktomystota cell plan includes intracytoplasmic membranes that separates the cytoplasm into peripoplasm, an outer ribosome-free space, and pielulosome, or riboplasm, an inner ribosome-containing space. 27. Membrane-bounded animoxosomes have been discovered in. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 5. Planktomystota animox genera, which perform anaerobic ammonium oxidation. 28. In the Planktomystota species Gemata obscura globus, a nucleus-like structure surrounded by lipid membranes has been reported. 27, 29. Compartmentalization is a feature of prokaryotic photosynthetic structures. 2. Purple bacteria have chromatophores, which are reaction centers found in invaginations of the cell membrane. 2. Green sulfur bacteria have chlorosomes, which are photosynthetic antenna complexes found bonded to cell membranes. 2. Cyanobacteria have internal thylakoid membranes for light-dependent photosynthesis, studies have revealed that the cell membrane and the thylakoid membranes are not continuous with each other. 2. Prokaryotic organelles and cell components. Organelle / macromolecule main function structure organisms. Anamyxosome. Anaerobic. Ammonium oxidation. Ladrin lipid. Membrane. Candidatus. Bacteria within. Planktomystota. Carboxysm carbon fixation. Protein shell bacterial. Microcompartment. Some bacteria. Chlorosome photosynthesis. Light harvesting. Complex attached to cell membrane. Green sulfur. Bacteria. Flagellum. Movement in external medium. Protein filament. Some prokaryotes. Magnetosome magnetic orientation. Inorganic crystal, lipid. Membrane. Magnetotactic. Bacteria. Nucleid. DNA maintenance. Transcription to RNA. DNA protein prokaryotes. Pilus. Adhesion to other cells for conjugation, or to a solid substrate to create motile forces. A hair-like appendage. Sticking out, though partially embedded into the plasma membrane. Prokaryotic cells. Plasmid DNA exchange. Circular DNA. Some bacteria. Ribosome, 70S. Translation of RNA into proteins. RNA protein. Bacteria and archaea. Thylakoid membranes. Photosynthesis. Photosystem proteins. And pigments. Mostly cyanobacteria. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. See also, edit. Core hypothesis. Ejectosome. Endosymbiotic theory. Organelle biogenesis. Membrane vesicle trafficking. Host pathogen interaction. Vesiculovacuolar organelle. References, edit. 1 AB Kefeld CA, Sawaya Mr., Tanakar S., Nguyen CV, Phillips M., BBM, Yates 2, August 2005. Protein structures forming the shell of primitive organelles. Science. 309, 5736 9368. Bibcode, 2005 SCI, 309936K. Site CX 10.1.1.1026.896. Do, 10.1026/science.1113397. PMID 16081736. S2 SID 24561197. 2 ABCDEFG Murat, Dorothy, Byrne, Megan, Kamalai, Arish, 1 October 2010. Cell Biology of Prokaryotic Organelles. Cold Spring Harbor Perspectives in Biology. 2, 10.000422. Do, 10.1101/perspect.a000422. PMC 2944366. PMID 207394111. 3 Peterson L., 17 April 2010. Mastering the Parts of a Cell. Lesson Planet. Retrieved 19 April 2010. 4 D. Gregorio Ma, 2005. From Here to Eternity, Ernst Haeckel and Scientific Faith. Göttingen. Vanden Hoek and Ruprecht. p. 218. 5 Bush Leo, 1888. Dr. H. G. Brons Klassen Ordnungen des Theorikswissenschaftlich. Dargestellten wird und Bild. Erster Band. Protozo. Drittabteilung, im Versicherungssystem der Radiolaria. p. 1412. Die Vakuolen sind demnach in strengem Sinn kein beständigen Organoder. Organule, wie Mobius die Organ der Einzelnen im Gegensatz zu denen der Weiseligen zu nennen Vorschlag. 6 Rider J.A., ed. February 1889. Embryology, the structure of the human spermatozoon. American Naturalist. 23, 184. It may possibly be of advantage to use the word organule here instead of organ, following a suggestion by Mobius. Functionally differentiated multicellular aggregates in multicellular forms or metazo are in this sense organs, while, for functionally differentiated portions of unicellular organisms or for such differentiated portions of the unicellular germ elements of metazo, the diminutive organule is appropriate. 7 Robin C. Pouchit G. Duval mm, Rhetor E. Tourneux F. 1891. Journal de l'anatomie et de la physiologie normale et pathologique de l'homme des animaux. F. Alkan. 8 AB Mobius K. September 1884. Das Steben der Einzelnen und der Weiseligen Tier. Wörtliches End. Betrachtet. Biologisches Centralblatt. 4, 13, 14389392, 448. Warrant die Fortpflanzungszellen. Der Weiseligen Tier unthätig fortalben bis sie sich lose außen, wandern und entwickeln, tritten die Einzelnen Tierort durch die an der Fortpflanzung beteiligten Lebensmassen Verkehr mit der Außenwelt und weil bilden sich die Verortsbesond Organule. Footnote on p. 448, die Organ der Heteroplastiden bestehen aus Vrenigten Zellen. The die Organ der Monoplastiden Nervertiden. Ausgebildet Teil einer Zell sind Schlag ich vor, sie Organule zu nennen. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 9 Walker, Patrick, 2009. Nuclear import of histone fold motif containing heterodimers by import in 13. Niedersächsische Staats- und Universitätsbibliothek Göttingen. 10 AB Keeling P.J., Archibald J.M., April 2008. Organelle Evolution, What's in a Name? Current. Biology. 18, 8R345-7. Bibcode, 2008 CBio, 18.R345K. Do. 10.1000.j.cub.2008.02.065. PMID 184306636. S2 SID 115200942. 11 Campbell Na, Rees J.B., Mitchell L.G., 2002. Biology, 6th ed. Benjamin Cummings. ISBN 978-0. 8053-6624-2. 12 Not TJ, Pet Salaki E, Faber P, Jarvis D, Fassner E, Plokow Ietzo, Craggs TD, Bazet Jones DP. Pawson T, Foreman KJD, Baldwin H, March 2015. Phase transition of a disordered Newage protein. Generates environmentally responsive membrane-less organelles. Molecular cell. 57, 59369447. Do. 10.1016/j.molchel.2015.01.013. PMC 4352761. PMID 2574769. 13 Banani SF, Liho, Hyman A. Rosen MK, May 2017. Biomolecular condensates, organizers of cellular biochemistry. Nature Reviews Molecular Cell Biology. 18, 528528. Do. 10.1038/NRM.2017.7. PMC 7434221. PMID 282250881. 14 Cormac DH, 1984. Introduction to Histology. Lippincott. ISBN 978-0-397-52114-2. 15 Brangwin CP, Ekman CR, Corson DS, Rabaska Hu Edge C, Garrick Hanny J, Julika F, Hyman. AA, June 2009. Germline P. Granules are liquid droplets that localize by controlled dissolution/condensation. Science. 324, 5935 1729 Bibcode 2009 SCI 324.1729b. Do. 10.1126/science.11720046. PMID 19460965. S2 SID 42229928. 16 Imanian B, Carpenter KJ, Keeling PJ, March-April 2007. Mitochondrial genome of a tertiary endosymbiont retains genes for electron transport proteins. The Journal of Eukaryotic Microbiology. 54. 214653 Do. 10.1015/j.1550-7450.2007.00245.x. PMID 1740315. S2 SID 203939495. 17 Mullis C, 2004. Theory of Organelle Biogenesis, a Historical Perspective. The Biogenesis of Cellular Organelles. Springer Science and Business Media, National Institutes of Health. ISBN 978-0-306. 47992-8. 18 Alberts B, Johnson O, Lewis J, Raff M, Roberts K, Walter P, 2002. The Genetic Systems of Mitochondria and Plastids. Molecular Biology of the Cell, 4th ed. ISBN 978-0-8053-3218-3. 19 FAARC, Newton G. L., Arik B., Overdenk Bogart T., Ali S. B., April 1984. Entomoeba histolytica, a eukaryote without glutathione metabolism. Science. 224, 4644-72. Bibcode, 1984 SCI, 224, 70F. Do. 10.1126/science.6322306. PMID 63222306. 20 Alberts B, Johnson O, Lewis J, Morgan D, Raff M C, Roberts K, Walter P., Wilson J. H., Hunt T., 2014-11. 18. Molecular Biology of the Cell, 6th ed. Garland Science. p. 679. ISBN 978-0815345244. 21 Badenow J.L., Mitsuma N., Beals P.L., Katsanis N., September 2006. The Ciliopathies, an Emerging Class of Human Genetic Disorders. Annual Review of Genomics and Human Genetics. 7, 12548. Do. 10.101146/7.080500505.115610. PMID 167228803. 22 Anderson P., Kedeshar N., March 2008. Stress Granules, the Tao of RNA Triage. Trends in Biochemical Sciences. 33, 314152, 10.1016/j.types.2007.12.003. PMID 182916557. 23 Psi Y, Sawaya Mr., Canon GC, CAIF, Williams EB, Heinhorst S., Kefeld CA, Yates 2, June 2007. Structural Analysis of XOS1 and the Protein Shell of the Halotheobacillus neapolitanus. Carboxysm. PLOS Biology. 5, 6E144 do. 10.1371/journal.bio.0050144. PMC 1872035. PMID 175185118. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 24 Ritter, January-February 1988. Contribution of new cryo methods to a better knowledge of bacterial anatomy. Annals de l'Institut Pasteur. Microbiology. 139, 13344 do. 10.1016/0769. 2609, 88, 90095-6. PMID 3289587. 25 Kamelia, Lee Z., Newman D.K., Jensen G.J., January 2006. Magnetosomes are cell membrane invaginations organized by the actin-like protein MAMK, PDF. Science. 311, 5758-2425. Bibcode, 2006 SCI, 311242K. Do. 10.1126/science.1123231. PMID 163735332. S2 SID 36909813. 26 Skifella, Gruska M., Favre D., Lenarau Diesel, Plitsko J.M., Stula D., March 2006. An acidic protein aligns magnetosomes along a filamentous structure in magnetotactic bacteria. Nature. 440, 7080. 110.4. Bibcode, 2006 Nature.44110s. Do. 10.1038/Nature 04382. PMID 162999495. S2 SID 4372846. 27 AB Lindsay, Mr. Webb, RI. Struss, M. Jetton, MS. Butler, MK. Ford, R.J. Fuest, J.A. 2001. Cell compartmentalization in Planktomystes, novel types of structural organization for the bacterial cell. Archives of Microbiology. 175, 641329. Bibcode, 2001 ARMIC.175413L. Do. 10.1007/S002030100280. PMID 11491082. S2 SID 219707003. 28 Jeton, Mike S.M. Niftrick, Laura Van, Struss, Mark, Kartel, Boran, Keltjens, Jan T. Opdenkamp. Huyub J.M. 1 June 2009. Biochemistry and Molecular Biology of Anammox Bacteria. Critical Reviews in Biochemistry and Molecular Biology. 44, 2365-84 do. 10.1080/104092309027227883. HDL 2066/75127. PMID 192478443. S2 SID 205694872. Retrieved 3 August 2020. 29 Fuest J.A., 13 October 2005. Intracellular compartmentation in Planktomystes. Annual Review of Microbiology. 59, 299328. Do. 10.1011.49.59.030804.121258. PMID 1591029. External links, edit. Media related to organelles at Wikimedia Commons. Tree of Life Project, Eukaryotes. Organelle databases. Categories, Organelle cell anatomy. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.